Milky stork, with the scientific name Miteria cinerea, is a large water bird 97 cm long. Their habitat types are forest, wetlands, nearby mangrove and marine coastal. The juvenile has greyish plumage, while the adult has white plumage, with a little portion of black on the wings and tail that is only noticeable when flying. The bill is long, yellow in colour, slightly decurved and rounded at the tip. The bird has a giant white stalk with reddish legs and feet. Random facts about them is that their leg will turn to dark red during breeding season. It feeds in fish ponds, sailing pools, freshwater marshes, and rice fields to consume crab, fish, and prawns. Mature and tall trees are crucial for nesting, while the peak nesting occurs between February and April. Unfortunately, the direct removal of their mangrove habitat activity has resulted in Milky stork being listed as endangered species on the IUCN Red List due to their current population having drastically decreased, with the estimation between 1,200 until 1,850 species left. To resolve this issue, captive breeding has been used by Zoo Negara. Captive breeding involves reproducing endangered or threatened species in regulated settings such as zoo without having to capture additional individuals from the wild. The objective is to guarantee the long-term survival of endangered species by increasing their number. Certain individuals may be returned to the wild when the population has stabilized to aid in the reintroduction effort or restore wild population. However, merely reproducing the bird is insufficient. Molecular tools such as DNA fingerprinting and genotyping and molecular sexing are applied to direct the breeding process to maintain their genetic health. Next, molecular tools are needed in captive breeding to avoid inbreeding, identify individuals and lineage. Some of the molecular tools utilized by Zoo Negara include DNA fingerprinting and genotyping. It's a straightforward and non-invasive procedure. A tiny sample is taken, such as a drop of blood or a feather. Next, microsatellite markers, which act as tiny genetic tags that indicate how similar or dissimilar each bird is, are used to collect and analyze DNA. Therefore, the zoo can determine the best possible pairs and assess genetic relatedness before making any breeding decision to keep the flock's genes strong and varied. Also, this can avoid inbreeding to protect the genetic health of the whole population. Another important molecular tool used in the captive breeding of Milky stork is molecular sexing. For breeding, it is necessary to identify their gender. Unfortunately, it is very difficult to distinguish Milky stork by appearance alone because they are monomorphic, meaning that the males and females have nearly identical appearance including size and feathers color. So scientists use molecular sexing, which involves testing the bird's DNA. They focus on a gene called CHD found on the sex chromosome in birds, which are Z and W chromosome. They amplify the DNA using PCR, then run it on a gel to examine the band pattern. Males will produce one band, while females will produce two bands. By knowing each bird's sex, the zoo can more efficiently manage the population, balance the male-to-female ratio, and arrange better breeding pairing.
Modern molecular tools such as DNA fingerprinting and microsatellite genotyping play a vital role in the captive breeding of Milky storks at Zoo Negara. These tools help conservationists monitor genetic structure and make informed breeding decisions. One major concern in small captive populations is inbreeding, which can lead to reduced fertility, poor hatch rates, weaker immunity, and genetic defects. By using genotyping, researchers can identify genetic relationships between individuals and avoid mating closely related birds, helping to maintain the population health. Another key benefit is preserving genetic diversity. Genetic variation is important for a species' long-term survival and ability to adapt to diseases or environmental changes. Genotyping allows scientists to assess genetic diversity and ensure that all genetic lineages are fairly represented in the breeding program. Lastly, DNA fingerprinting helps identify hybrids or misidentified individuals. In captivity, Milky storks may interbreed with painted storks, producing hybrids that are hard to distinguish visually. Molecular tools ensure only pure milky stocks are used for breeding and reintroduction, protecting the species' identity and ecological role. Next, molecular sexing using the CHD gene and PCR is an important tool in the conservation of monomorphic birds like milky stocks, where males and females look alike. Traditional sexing methods based on appearance or behavior are often inaccurate and can lead to incorrect pairings or poor breeding outcomes. This technique allows accurate identification of each bird's sex, which helps form compatible pairs and improve breeding success. It ensures a balanced number of males and females, leading to better clutch production and more efficient use of conservation resources. Additionally, knowing the sex of all individuals support demographic planning, especially when preparing birds for reintroduction into the wild. Accurate sex ratios help maintain healthy, sustainable populations both in captivity and in natural habitats. Overall, the endangered milky stocks were successfully breeding through the conservation program at Negara, which shows significant increase in the number of milky stocks. Over 100 milky stocks in total managed to breed, which is considered a huge achievement for such an endangered species. Some of these birds have been released into the safe wetland areas at Kuala Selangor and Kuala Gula to restore natural ecosystems and rebuild wild populations. It is a huge step in the right direction of their natural population growth. Thus, it is possible to reverse the decline of a threatened species. Even though the breeding program had great success, it also faced some tough challenges, especially in the early years. One of them was that many of the hen-raised milky stocks chicks started developing bone deformities, especially in their legs, known as metabolic bone disease caused by imbalances in calcium and vitamin D. Unfortunately, the deformities were so severe in some chicks that they could not walk or stand properly. Several of these chicks had to be put down due to their irreversible condition. As a result, many had to be culled to prevent further suffering.
A comprehensive solution to bone defect in chicks involves integrating genetic and nutritional approaches. Scientists can use genetic screening to detect mutation or variation in head-related genes associated with skeletal development. Early identification enables selective breeding programs that reduce the incidence of defects. At the same time, tracking chick growth and development provides critical data to adjust and optimize breeding protocols, ensuring chicks receive adequate levels of calcium, phosphorus, vitamin D, and other essential nutrients. This dual strategy, accurate genetic monitoring and nutrition, can significantly reduce bone abnormalities, improve animal welfare, and enhance the overall health, productivity, and sustainability of poultry operation.